much for the introduction. I'm not sure where my slides are, but I can at least get started by saying that this is a continuation of the non-genetic aspect of these proposals by focusing on a global understanding of proteomic and metabolomic profiles in health and disease. So this is moving beyond just looking at the host genetics and looking to try to understand the intersection between the host and the environment and looking at the impact on circulating proteins and metabolites. So encompassing further down on the omics spectrum by trying to look at both proteomics and metabolomics. So we know that there is inter-individual variation in protein and metabolite concentrations by age, by environmental exposure, including diet and lifestyle, and by healthy versus disease states. We also know that there are many examples of protein and metabolite biomarkers that are associated with disease risk. And I just wanted to give you a little bit of an example by focusing on the branched chain amino acids for a minute, looking at their relationship with lifestyle factors as well as with disease risk. So the branched chain amino acids, leucine, isoleucine, and valine, are associated with adiposity. And in this figure from a paper by Reitman and colleagues, found an association between circulating valine levels and visceral adipose tissue, with the circles representing data points from lean individuals and the pluses representing data points from obese individuals. And the correlation here is about 0.48. Then there have been several studies that have shown that circulating branched chain amino acids are associated with subsequent risk of diabetes. And I'm showing you here some results from Tommy Wang and colleagues who looked at this relationship in the Framingham offspring study as well as the Malmo diet and cancer study. And what you can see here going across the results is for each of the individual branched chain amino acids, individuals who had higher circulating levels were at a higher risk for subsequent diabetes, such that people in the fourth quartile had about threefold higher risk compared to people in the first quartile. And this is after accounting for BMI and fasting glucose in these results, and also in other results accounting for other indices of insulin resistance as well. And then I also wanted to give just a glimpse of possibilities extending this in the future in the context of something like the IHCC. It's possible to be able to look at these biomarkers with more rare outcomes. So this is looking at branched chain amino acids and pancreatic cancer risk, done by one of our colleagues, Brian Wolpen, who found that the three branched chain amino acids, and I think this figure is actually flipped, but at any rate, it's leucine, isoleucine, and valine showed up as being significantly associated with risk of pancreatic cancer. And they also were able to look at the timing between blood collection and diagnosis and found that the relationship was strongest for the bloods that were collected 2 to 5 years prior to diagnosis, suggesting that these were actually markers of occult disease. So it's another interesting way that we can get at a better understanding of the biology of the larger context of a consortium. So the motivation for this comes from wanting to understand the impact that, or the implications of diverse populations on proteomic and metabolomic profiles to be able to understand how these omics profiles result in health and disease etiology by taking into account the different demographics of age and race, ethnicity, et cetera, thinking that eventually this could lead to precision medicine approaches to prevention and treatment by population. So we know that adiposity and metabolic abnormalities are driving some of the increase in non-communicable diseases around the globe. And diabetes is really the clinical manifestation that is most directly related to these underlying issues. We know that diabetes is increasing worldwide. We also know that rates differ by race and ethnicity. And in fact, the relationship between BMI and diabetes differs across the globe. So we're hoping that this would lead to a better understanding of the pathway to diabetes and mechanisms underlying the link between obesity and diabetes, and can help inform some of our understanding of how adiposity and metabolic dysregulation might be impacting other diseases as well. So I also think this is a public health opportunity to interrupt the cycle of increasing incidence of diabetes across the globe. So leveraging the unique diversity of the cohorts in the IHCC, our aims include, one, to investigate differences in proteomic and metabolomic profiles across populations, spanning racial, ethnic, geographic, and SES groups. Two, to investigate how lifestyle factors influence proteomic and metabolomic profiles. And then three, to examine the role of these omics profiles in the development of chronic disease using diabetes as a pilot. So at the outset, we propose to conduct metabolomic and proteomic profiling in 2,000 disease-free individuals selected across a globally representative set of cohorts. And within any of these cohorts where this is possible, to also select up to 1,000 diabetes cases that are diagnosed after the time of blood collection, and then match them one-to-one to controls that would make up part of this, a subset of the 2,000 disease-free individuals. So in terms of proteomics, we would propose to use this Olink platform, which uses a proximity extension assay, which uses a dual recognition, 2 antibodies coupled with DNA for identifying. It's highly multiplex, comes in modules that can be selected for a total of about 1,300 proteins that are assayed with relatively rapid throughput. And we've pilot tested this platform in our own cohort samples from the Nurses' Health Studies and have seen excellent results so far. And then for metabolomics, we propose using the Broad Institute's platform that's been developed by Clary Klisch, which is an LCMS-based metabolomics semi-targeted platform with 4 different platforms with different extraction methods and positive and negative ion mode. And the figure on the right is a little bit out of date, but at this point, it's upwards of 500 metabolites that have been identified. But we also get thousands of unidentified peaks, which I think will allow for a rich data source to be continued to be interrogated going forward in the future as these peaks continue to be identified. So in terms of analyses for both proteins and metabolites, we can break these down in a number of ways, obviously looking at the individual proteins and individual metabolites. We can also think about different classes. So for proteins, we can look at kinases or growth hormones, growth factors or hormones. And for metabolites, we could look at classes, amino acids or subclasses of lipids. But then, perhaps even more importantly is to be able to take a network approach and look at the omics profiles in their entirety and understand how these intersect together. So taking a few different approaches with the proteins and metabolites, we can look to try to understand how demographic, geographic, and lifestyle factors impact these proteomic and metabolomic profiles. And then understanding how the proteomic and metabolomic profiles are related to subsequent onset of diabetes, and then taking the intersection and trying to understand how these demographic and lifestyle factors intersect with the proteins and metabolites and impact risk of disease as well. So we anticipate this would be a 3-year project, and the activities would include defining the data set, so trying to pick demographic, lifestyle, environmental variables, and I would propose as part of this pilot project, we know how complicated it is to harmonize data, that we could select a sort of a manageable subset to start with, and then creating a central data repository, although using Anthony's terms, it would be a federated data repository where we can then harmonize the data together, which is obviously important for this step, but we also know how complicated this is, having participated in many consortia before. Then selecting participants, actually getting the proteomics and metabolomic profiling completed, and then doing statistical analyses. So we've outlined some potential policy issues, which I'm sure are not unique to this particular proposal. But one sort of overarching question, thinking about country-specific policies, is it possible for blood samples to be shipped to a central laboratory to conduct the profiling? Another challenge, which goes for any of these proposals as well, is how well can data be harmonized across cohorts, and how much, if we go for the least common denominator approach, how much information do we lose by doing that? And then thinking about what is the model for a central portal for the federated data, which, as Anthony had proposed, could be something as part of the underlying infrastructure that we can use all together. So in brief, thinking about the scientific processes required, it would be accessing archived samples, shipping them to central laboratories for proteomic and metabolomic profiling, preparing the data sets within the cohorts, as well as trying to understand how the federated data set works, and then conducting analyses. So year one, we anticipate focusing on some relatively critical tasks, including understanding how to design the variable harmonization and what sort of variables we would pick as a starting point of a kind of smaller, manageable subset. Designing and figuring out how to use this central repository for the data deposition, selecting participants, designing QC pipelines and allocating QCs across the different cohorts, and then, of course, accessing blood samples within each cohort. Then year two would focus on getting those blood samples to the assay laboratories and working on getting the harmonized data from individual cohorts to the federated model. And then year three, we would be receiving the assay data back, sending it through QC pipelines, and then uploading to the central repository, and then move on to analyze the data and prepare manuscripts. So the dependencies, I think we need to consider what the country-specific policies are on biospecimens and how we as a consortium can fit a project like this within the bounds of what some of those policies are. And then the timing and access to blood samples can be probably cohort and country specific. And so we need to consider how this would impact the proposed work as well. In terms of cross-cohort needs, our goal is to look at global differences in proteomics and metabolomics, which requires the representation and distribution across different ethnicities and geography. In terms of participating cohort tasks, it would include selecting participants, aliquoting blood samples, shipping them, and then preparing the data that would need to be used in a harmonized way for this project. So we proposed cohorts across the US, Africa, Asia, India, South America, and Europe for this pilot project. And in terms of resources, we've outlined an estimated budget that would probably be upwards of $2,000 per sample for accessing the blood samples and then doing the proteomics and metabolomics profiling. And in terms of personnel, we would foresee having postdoctoral fellows and junior faculty involved in helping to lead this effort. There's going to be required investigator time within each cohort that needs to be supported to be able to contribute data and contribute to the collaboration. Investigator time for overall leadership of the project, and then investigator and programmer time to work on this federated data model, which I think could be in concert with other projects as part of the IHCC. And then both within cohorts and centrally, we would need investigator and programmer time to help prepare the data, as well as receiving the omics profiling data and processing the data. So this is sort of a large-scale summary of the resources that would be required to achieve this pilot project, but it would be achieving not just a demonstration of feasibility, but also generating data and answering scientific questions, all encompassed within this project. And with that, I'll thank you and happy to take any questions.
Thank you. We have time for one question.
Usha Menon, UCL. You didn't put anything about the material transfer agreements that would have to be created between each of the institutions, and that would take some time.
Yes, you're absolutely right. In terms of the data use agreement and material transfer agreement, you're right. It's an important consideration.
Terry?
We've heard a lot about those unnamed peaks in metabolomics, and I was just wondering, has anybody actually identified any of those? And because I haven't heard that's been an effort that's moved forward.
So there have been some examples from Clary Klish's laboratory, and in fact, one of the strong metabolomic predictors of diabetes is DMGV, which originally was an unidentified peak that showed up as being strongly associated with diabetes, and going back and finding that it was also associated with fatty liver. And then working together with a few different cross-cutting teams, went back and identified that metabolite. So it's right now, I would say it's a process of picking metabolites that are showing up as of interest and trying to then identify them. And he has an ongoing effort to continue to identify these. So I think it's a moving target, but it's an ongoing process.
Just one information. How much importance will be the information on freeze and thaw cycles? Because many of the cohorts will not have that information. So is it essential, or it's okay if you don't have that information?
So that's a great question. And one of the things that we are always careful to do in the Nurses' Health studies, our samples are not collected in ideal conditions. And we are always sure to pilot test what effects those differing conditions have on the metabolite results. So for instance, our samples were collected and shipped overnight before they came to the lab to be processed. And we know that we can't measure glucose-related metabolites, but I think those data are important for us to know how to utilize the metabolomics data that we get back. In terms of freeze-thaw cycles, I think Clary has done actually a lot to understand the impact of freeze-thaw cycles on the metabolomics. And I think that's a case where we could consider which metabolites in particular would be sensitive to that, to understand that aspect when we're analyzing data where we either know that there are multiple freeze-thaw cycles or that we don't know at all. It's an important point.
Thank you, Heather.
Thank you.